Unit C, cycling of matter in living systems, also known as the biology unit of Science 10. This is going to be a little bit different from what we're used to in Science 10 in that biology does have a reputation of being pretty heavy in the memorization area, which there definitely are aspects of biology that are memorization heavy. There's also some places in here where we will be doing calculations, especially when we're talking about the microscope and the efficiency of cells and whatnot. So we're not done with math yet, but the majority of stuff in this unit is going to be memorization based. So we're going to be talking about some early scientists that contributed to biology. We're going to be talking about parts of the cell, parts of the plant tissue, et cetera, et cetera. Okay, so this is going to take us in the neighborhood of around 15 to 20 classes to get through, depending on how quickly we move. And we will be having a quiz after section C1 and C2. So a C1, C2 quiz combined, and then we'll be having a unit exam. We're also going to be doing a microscope lab where you're going to be getting some practice doing some wet mounts and stuff. So we're going to be moving pretty quickly. And yeah, there's not a ton of assessment in this particular unit, especially when compared to physics and chemistry. Okay, so you have your note packages. I've bolded and highlighted the words on my PowerPoints so that it's fairly easy for you guys just to kind of glance up and fill these in. Again, biology, lots of terminology. So, you might want to have a separate maybe duotang or binder where you're just writing vocabulary down. Flashcards help, going over your notes and highlighting that sort of thing, because there is a lot of material here. Okay. Okay, so our first few sections here deal with the microscope, microscope advancements, and microscope technologies. So the microscope is an incredibly useful tool in biology, because a lot of the stuff that we deal with in biology is not visible to the naked eye. And even the stuff that we do see is made up of very tiny objects like the cell. Okay, so it's really handy to be able to have a tool to really look closely at some of the stuff we're studying, because all living things are made up of smaller components, okay, namely the cell. And then, of course, the cell has structures inside of it that are important. So the microscope, incredibly important. The microscopes that we have today, they have quite a history. So really, there are two main kinds of microscopes. There's lots of different forms of these microscopes, but essentially microscopes fall into two categories. Okay, category number one, and this is what we will be using in the lab. And if you go to university, your first couple of years in biology labs, you'll be using this microscope as well. And it's called a light microscope. So light microscopes, like the name says, uses a light source, okay, visible light, and a system of one or more lenses to magnify an image. So early microscopes, they used only one lens. The microscopes that we use primarily in high school and in your early undergrad classes in university will be light microscopes, and they will have multiple lenses. Okay, and the image that we get using one of these kind of looks like the one that's up on the screen there. So you can see the different cells. These are plant cells. They have a stained cell wall, and we can see the nuclei, and that's about it. So really expensive light microscopes, we can get, maybe in the 1,000 type magnification. The microscopes that we're going to be using in school get us to the 100, X magnifications ish, like 1, 2, 3, 400 times. We need other microscopes if we want to see even smaller objects. So the other type of microscope or class of microscope are called electron microscopes. And instead of using light, they use a focused beam of electrons. And these electrons bounce off of specimens and they're collected by a special collector. And we need computers to construct images. So you can see there, that's an ant, right? Your everyday garden variety ant. And you can see that we can see it in incredibly precise detail. All right, so that's a picture of a fairly small electron microscope, and you can see that it takes up, a fairly sizable office. Right? We need something to create the electron beams. We need a vacuum chamber, and then we need computers to construct these images. So these are incredibly expensive, and you have to be specialized in order to use them. You need special training, and the specimen has to be preserved in a very special way. So the naked eye, we can see almost down to, again, you guys have younger eyes. I probably couldn't see this, but you could see down to about 100 micrometers, anywhere from about millimeter to, or a meter, sorry, down to 100 micrometers. A light microscope gets us down into the nanometer range. And then a light electron microscope gets us down way, 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 way down to hundreds, tenths, hundreds, and even thousands of nanometers. So there are some electron microscopes that are allowing us to see down to the atomic level. Right, like there are actual pictures I saw on Google when I was making this presentation, that they have actually developed microscopes that can see down to the atom and molecular level. Now, as far as light microscopes go, there are kind of two types of light microscopes. Again, remember, a light microscope is any combination of a light source and a lens or lenses. So a simple light microscope uses just a single lens and just the ambient light in the room as a light source. So a magnifying glass would be an example of a simple light microscope. It's got one lens that makes objects larger, and it just uses the light from the environment. Okay, here's a little bit more sophisticated, simple microscope. This is from around 1871, when microscopes were just starting to be refined. Okay, again, it only has one lens up here, and here's the light source. It's basically a mirror that just reflects light. Okay, so those are what are known as simple light microscopes. A compound light microscope consists of two or more lenses, and usually has a light source like a light bulb. Okay, so in this case, we have a lens here in the eyepiece. Okay, so that would be one lens. And then we have a lens down here as well. And the light source is down here. And this would be either battery operated, or you would plug this into an outlet and it would have a light bulb in there, and the light would shine up and illuminate your object that you're looking at. And then the lenses together would add their magnifying power to magnify the image. Okay, so this is known as compound because we are using two lenses, and we're using the power of both of those lenses combined to magnify the image. Now, microscopes, they go back nearly 500 years. So the very first compound microscope was invented around 1595, and it was invented by a father and son duo, and they were actually lens grinders. So they made lenses for eyeglasses, and they found if you combine these and you look through these fine lenses, you could actually magnify objects quite a bit, and you could see things in quite a bit detail. Okay, so the compound light microscope has been around for about 500 years. Now, the lens that you look through is known as the ocular lens. Okay, so this is the lens that's closest to your eye. And the lens further away, this is known as your objective lens. And even today, with today's microscopes, we still use the same terminology. Okay, ocular is the lens that's closest to your eye, usually the one you're looking through, and then the objective is closest to your specimen. So this would be a compound microscope because we have multiple lenses. Now, Robert Hooke used a 3-lens system in his microscope. So he was getting even more magnification. And he used a special lamp, and then he took the lamp and he projected the light through a water flask to make that light even more intense, to really illuminate his specimen. So he's really able to get a bright light on his specimen, and he used 3 different lenses in his microscope, and he was really able to magnify objects quite a bit. Now, Robert Hooke is very famous, and you might want to put a star beside this. Robert Hooke was really famous in that he actually coined the term cell. So he was the first one to describe a cell using that name. And the reason he did this is because when he looked at plants, he found that plant tissue was made up of these little objects, and he thought that it looked kind of like, well, we think of prison cells, right? But back then, monks lived in these little tiny rooms that were called cells as well. And he was familiar with these, and he said, oh, they look like these little rooms. So he termed them cells. Now, he didn't know what they were used for. He didn't know what was contained inside of them. He just looked at these plants and said, oh, look, they're made of these little rooms, essentially. Okay, and this was in the late 1600s. And these are actual drawings by Robert Hooke that he did of the objects that he was looking at. Now, Anton van Leeuwenhoek, he used a simple light microscope with a very, very, very good lens. So lens technology really advanced over a few hundred years, from the late 1500s up until the 16 and 1700s. So the lenses got really good, and he was able to use a single lens to observe things like pond water, pollen, and sperm. And he did lots of detailed drawings. And he called these little creatures that were not visible to the naked eye, but through his little microscope, he could see these things swimming around, and he called them animalcules. Animalcules. That's a mouthful, that word. Animalcules. So little tiny animals swimming around in these samples. And again, he was an excellent artist, and he was able to document some of the things that he saw. Now, we will be using a compound light microscope in Science 10. We will go to the lab later on, and you will have to use the microscopes to view some specimens. You'll have to do some drawings and some calculations. So this microscope right here, very similar to the one that you will be using. It will have a light source, and it will have an ocular lens as well as an objective lens. So in order to use the microscope in the lab, you're going to have to be familiar with a few things. You're going to have to be familiar with the structures and functions of the parts of the microscope, how to handle a microscope, calculating magnification, some unit conversion, And then there's some other microscopy calculations that are involved in dealing with things like field of view, etc. Okay, so this is fair game, ladies and gentlemen. I will be giving you on your quizzes and exams, I will be giving you a microscope like this, and I will be asking you to label the parts of the microscope. So you have to be able to replicate what's on the screen here. You have to be able to label a microscope. So I'm going to give you a couple of minutes here to write these down. All right, so hopefully you've labeled those parts. Again, this is fair game for any tests, quizzes, exams that I give to you, and you need to be familiar with the terminology for your upcoming lab. So essentially, these parts of the microscope are important for a number of different reasons. Obviously, number one, this is the ocular lens. This is where you look through in order to view the specimen. Okay, the specimen is held here on the stage. So number 9 is the stage. This is where the specimen goes, and there'll be clips on the stage to hold down your the slide that you might have your specimen on. Okay, so those are called stage clips. The specimen lies on top of the stage, and the stage can go up and down in order to focus the image. Underneath the stage, there's something known as a diaphragm, and this allows certain amount of light through, and it can focus light. We can put different filters in there if we use different stains on our objects. Number 7 is the light source. So light shines up through the condenser and the iris and the diaphragm, and there's a little hole in the stage that light can come through, and it can shine up into these lenses right here, which are known as the objectives. Now, the microscopes that we're going to be using in our lab, we're going to have 3 different objectives, and they will magnify the image at different amounts. Okay, we're going to have 2 knobs over here. One is called the coarse adjustment, one is called the fine adjustment. The coarse adjustment is used under low power, and it really moves the stage up and down significantly. Okay, you never want to use coarse adjustment if we're using medium or high power objectives, because we could smash the specimen up into the objective, and that could crack the slide, or it could crack the lenses. Okay, for that we use what's known as the fine adjustment, and that's number 4. Okay, so 3 is called coarse adjustment, it's the larger knob. Number 4 is the fine adjustment. Number 6 is the base, and number 2 is the arm, and that's what you use to carry your microscope. Always carry your microscope using 2 hands. So one hand on the arm, one hand on the base. Okay? Now again, you'll get some practice using these. We don't actually use them very much at all. I don't use them at all in Bio 20 or Bio 30, but you will use them in university quite a bit if you decide to go into any kind of biology related career. Okay, so as I just mentioned, always, always, always carry the microscope with 2 hands if you're ever transporting it around the lab. Okay, one on the arm and one on the base. We do not use brown paper or Kleenex to clean the lenses, only a special type of paper called Kim wipes. Okay, it's a special type of paper that will not scratch the lenses. Don't ever allow the lens to come into contact with the slide. Again, we could crack the slide or crack or scratch the lens. Don't try and fix the microscope yourself. In fact, we have a special person that comes in once a year to fix our microscopes. Just let me know if there's something wrong. If I can fix it, I will, but otherwise we set it aside and let an expert do it. Always, 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 always start your focusing under low power. Okay, and then once it's focused, then you can go to medium, and then you can go to high. Never use the large knob, the coarse focus, under high power. Like I said, you can wreck your specimen and you can rec the lens. When you're done, you rotate back to low power for the next person. You remove the slides, turn the microscope off, take the power cord off, and then we have dust jackets that go over top the microscopes. We just got all brand new microscopes this year. So we want to take care of them so they last 20 or 30 years. So there are a number of calculations that we have to do in biology when we're dealing with microscopes and viewing specimens under microscopes. The first thing that we need to know is the magnification of the image that we're looking at. So magnification tells us how much larger the image is than in real life. So for example, when we're looking at this little critter right here, the 40 times tells us that the image is 40 times larger than the actual animal. In this case, 100 times greater, 200 times greater, and 400 times greater. Now notice as the magnification increases, the structures that we see, the more of them are revealed. We're able to see more detail of these fine structures, but we're able to see less of the actual organism. So magnification is great in that it lets us see these little structures, but we can't see as much of the organism. Now, how do we determine this number here, this magnification? Well, it's a simple formula. Okay, magnification is equal to the power of the ocular lens multiplied by the power of the objective lens. Now, remember, the ocular lens is what we're looking through, and the objective lens is the lens that's closest to the specimen. So this would only be the case when we're using multiple lenses in a compound microscope. Okay, so a little bit of practice here. Generally, the ocular lenses of the microscopes that we use here at O'Leary are 10 times. So you'll notice that that doesn't change. What does change is the objective lens. So under the lowest power objective lens, it's only about 2.5 times magnification. So the way that we figure out the total magnification is we take the ocular and we multiply it by the objective. Now, all these formulas you can find on the very last page of your data booklet. You do not have to memorize these formulas. You just have to remember that you can find them on the last page of your data booklet, and you'll always be given your data booklet for any tests or quizzes. So the ocular lens is 10 times. Okay, multiplied by the objective lens, in this case, is 2.5 times. That's under low power. So we have a total magnification of 25 times. Okay, just as simple as that. I said there'd be math, but this is not by any means calculus or trig or anything. This is biology math, so not really difficult. You just have to know when to use it. Okay, so again, same kind of question here, but now we're looking for the magnification under high power. So the ocular remains at 10 times, and we're multiplying it by the high power objective, and in this case, it's 100 times. So our total magnification using these two lenses would be 1,000 times. Okay, 10 times 100. So very simple. Now, as I mentioned, what we can see under the microscope changes depending on our magnification. Now, what we see under the microscope, this is known as our field of view or field diameter because it encompasses the diameter of the lens, so the image that we're viewing through the lens. So in this case here, we're seeing some red blood cells, we're seeing a white blood cell, we're seeing a platelet here, but we're seeing a lot of these cells. So this would probably be under low power. So the field of view is everything you can see through the lenses. This is also known as the field diameter. So field of view is equal to field diameter. Now, the way that we determine this is we actually have to use a device to determine the field diameter under low power. Now, this device sometimes is built into the actual microscope, and sometimes we actually have to place a ruler underneath the objective lens on the stage and manually determine the field diameter. So remember, the field diameter is the total distance from one end to the other of what I can see under the microscope. So these are 1 millimeter markings. So in this case, we can see 1 millimeter, 2 millimeters, 3 millimeters, 4 millimeters, 5 millimeters, 6 millimeters, and part of a millimeter. So I can't really determine what this is. It's kind of guesswork. I'm going to say that this is almost a full millimeter. So maybe this is anywhere from, I don't know, 0.7 to 0.9 millimeters. This is an estimate. So I'm going to estimate that this field diameter or field of view is about 6.7 millimeters. Now, if you put 6.9 or 6.8 or even 6.5, you would not be wrong here. Okay, so this is under low power. So I go in, I determine what my field diameter is by literally measuring with a clear ruler. Okay, and I determine what the field diameter is under low power. Now, why under low power? Because once we shift to medium and high, our field diameter gets smaller and smaller, and sometimes it would be so small that we wouldn't actually even be able to see one increment. So how do we determine the field diameter under medium and high power? We have to use a little bit of math. Okay, so just a little bit of practice here. What would the field diameter be in this particular case? Well, these are 0.1 millimeter increments, and this is a full 1 millimeter. Okay, so this is 0.1, 0.2, 0.3, 0.4, 0.5, 0.6. So I definitely have 1.6. And it looks like I have about a half an increment here. So this would be 1.65 millimeters. Now, in this case here, our ruler tells us that we have 1 millimeter, 2 millimeters, 3 millimeters. We have a total of 4 full millimeters. And then this looks like about, again, this is estimation, about 0.1 or 0.2. So I'm going to estimate that the field of view is 4.2 millimeters. Now, again, this is not an exact science. If we had more precise measuring tools, we could get more precise measurements. But with the tools that we have, I'm limited to the precision. Okay, so 4.2 millimeters would be fine. Now, as I mentioned, as we increase magnification, so as magnification increases, the size of what you are seeing decreases. So in other words, as magnification goes up, our field of view goes down. Notice how many cells I can see along this field of view here, right? Probably like 20 or 30 cells I can see across here. Now, as we jump up to 100 times, look, it's only like 1, 2, 3, maybe 4 cells across, right? And then as we jump up to 400 times, I can only see maybe, I don't know, what, 1, 2, 2.5 cells across. So my field of view is getting smaller and smaller as my objects are getting bigger and bigger. Now, sometimes under high power and medium power, we can't actually measure because we can't actually see the lines of the ruler. Okay, under medium and high power. So we have to use a little bit of math in order to determine our field of view under those powers. Now, again, this can be found under the section C on the last page of your data booklet. You do not have to memorize these formulas. Okay, so look here. These are millimeter markings. Here I have 1, 2, 3, 4, and a little bit, right? That's under low power. Then under medium power, this is only like 1.5. Now, under high power, I don't even know how many millimeters this is because I can't see the ruler fully under the high power objective. So I need to figure out using math, what is the field diameter under high power. So let's practice. So what I'm given here is I'm given my low power objective. Okay, so under most microscopes, it's anywhere from 2.5 to 4. It depends on the microscope. You'd have to look. So the low power objective in this case is 4 times. The high power objective in this case is 100 times. And the field diameter under low power is 4 millimeters. Now, where did I get this? I measured this. Okay, I physically measured this in the lab. So now I just take these numbers and I plug them into this formula. Okay, so what I'm trying to determine is the high power field diameter. Okay, that's my unknown. So I'm going to call this X. Okay, the low power field diameter is 4.0 millimeters. My low power magnification is 4 times, and my high power magnification is 100 times. So I can write the formula as X divided by 4.0 is equal to 4 over 100. And I simply just multiply this side by 4.0. That's going to cross this off. And I have to do the same here. Multiply by 4.0. This gives me 16 over 100, which gives me 0.16 millimeters. Okay, and that's the field diameter under high power. Much smaller than the 4 millimeters that I can see under low power, right? Now, in this case, I'm being asked for the medium field diameter. And again, if you look at your data booklet, on the last page of your data booklet, it gives you this formula right here with high power. Now, what am I going to do? I'm not being asked for high power. I'm being asked for medium power. Well, it's just as simple as just replacing high power with medium power. Okay, so notice now I've just put in medium power here, and I've put in medium power here. Okay, and that's a little different than your data booklet, but I can do that, right? It's just a ratio. So again, I'm looking for the medium power field diameter. That's my X. Low power field diameter is 4.0 millimeters. Low power magnification is 4 times. Medium power magnification is 40 times. Okay, so X over 4.0 is equal to 4 over 40. X is equal to 16 divided by 40. So X is equal to 0.4 millimeters. Okay, and that's the field diameter under medium power. Okay, this is X. Now, once we know the field diameter, we can actually estimate the actual size of the objects that we're viewing. Okay, again, I'm sounding like a record that's broken. It's skipping because I'm repeating myself over and over, but you can find all of these formulas on your data, in your data booklet, on the last page of your data booklet. Okay, so we don't have to memorize these. So the actual size of an object is equal to the field diameter divided by the number of objects estimated to fit in the field of view. So for example, the field diameter under high power in this slide was determined to be 300 micrometers. What is a micrometer? A micrometer is 1,000th of a millimeter. So if I'm given millimeters, the way that I find the number of micrometers is that I times by 1,000. So 1 millimeter times 1,000 is going to give me 1,000 micrometers. Okay, and the opposite is true. If I'm given micrometers, okay, so in this case, 300 micrometers, how do I get millimeters? I divide by 1,000. Okay, so this would be equal to 0.300 millimeters. Okay, millimeters, sorry. So millimeters are quite a bit bigger than micrometers, 1,000 times bigger. So in this case here, I have 300 micrometers across my field of view. And I want to know how big are those plant cells in real life. Okay, so what I do is I count the number of objects that fit across the screen. In this case, here's one cell. Right? There's one cell. And then here's two cells. So about two cells fit across the screen. Okay, so the actual size would be equal to the field diameter, which is 300 micrometers, divided by the number of objects that fit across, which is two cells. So the actual size of these cells is about 150 micrometers. Okay, last but not least, scale. So scale compares diagram size. So when we're working with scale, we actually have to have a diagram or we have to draw a diagram. And what we're doing is we are comparing the diagram to the actual size of the image that we're, or the specimen that we're looking at underneath the microscope. Okay, so scale tells us how big our image is compared to whatever we're looking at. Now, again, just like every other formula that we've talked about in this lecture, we can find this on the very last page of our data booklet. Okay, so scale is equal to the size of the diagram of the object compared to the size of the actual object. Now, in the last set of calculations, we were able to estimate the actual size of the object once we know what the field of view is, right? And again, another name for field of view is field diameter. So once we know how big the field diameter is, then we can estimate at how big the object is, we draw it, and then we compare our drawing to the size of the object. So in this particular slide, what I have is I have a bunch of blood cells that are being viewed under a microscope, and it's telling us that the actual size of the object here, so if we take one blood cell, the actual size of the blood cell is 0.007 millimeters, okay, or 7 micrometers. Remember, you guys, micrometers looks like this, and there are 1,000 micrometers in 1 millimeter. So 7 micrometers is equal to 0.007 millimeters. Now, why is this important? Because I always have to use the same units when I'm comparing a drawing and the actual size of whatever I'm drawing. So here we can't use micrometers and then centimeters. Okay, right? My drawing, I measured at about 2 centimeters. Okay, so if I take it from here to about here, that is roughly 2 centimeters. Okay, now I can't use different units. I can't use micrometers and centimeters, so I kind of convert them to something in the middle. So each centimeter is equal to 10 millimeters. So 2 centimeters is equal to 20 millimeters. 7 micrometers, I divide by 1,000 to get millimeters. So now both of my measurements are in the same units. Now, could I have converted the drawing to micrometers? Sure, but I'd have a huge number. Okay, so now all I do is plug these numbers into the formula. So the size of the diagram of the object, that's this, okay, 20 millimeters. The actual size of the object, okay, I calculated at 0.007 millimeters, okay? And all I do is just divide the two numbers to get the scale. So 20 divided by 0.007 gives me a scale of 2,857 times. So what this is saying is that my drawing is 2,857 times larger than an actual red blood cell. Now, if we want to be consistent, we can convert this to one significant digit because this has one significant digit in it. So we could put this into scientific notation and just call it 3 times 10 to the third times. Okay, that is my scale. All right, so lots of calculations there for you to keep in mind. Please look at page 6 of your note package, also known as the Minions worksheet. This will give you some practice in doing all the calculations that we just went over in the previous lecture. And again, you will be using these on your microscope lab, as well as they're all fair game for any quizzes or exams in this unit.